Hello everybody. Thank you so much indeed for joining this, and there may indeed be more people who will join us. So it's a real great pleasure to do the first of what I hope becomes quite a lot of presentations on the research for development that we are sponsoring through the Current and Emerging Threats to Crops Innovation Lab, which is at Penn State University. And the first person we chose for this is Everlyne Wosula. Everlyne is from Kenya originally and lives in Tanzania and works for IITA. She has done her undergraduate work at Jomo Kenyatta University and has a PhD from Louisiana State University. And we were really keen to have Everlyne be our presenter here because many of us in various countries through the PlantVillage program are extremely interested in cassava diseases and particularly the whitefly vector. And the work that we're sponsoring from the Current and Emerging Threats to Crops Innovation Lab to Everlyne and her colleagues like James Legg, with whom we've worked for many years, is really, really promising. And so we've asked Everlyne to come along and to speak about both that work, but also the broader research agenda that she has for controlling whitefly, which of course is an incredibly big problem and perhaps one that will become bigger under the context of climate change. So Everlyne, you have a great audience here, 65 people I see. People feel free to put your introductions in the chat. If there's any questions that pop up, put them also in the chat, and we'll address them after Everlyne's presentation. But over to you, Everlyne. It's a great pleasure to have you here. So please take it away. Thank you.
Thank you, David, for the introduction, and welcome all to my presentation. So the title of the presentation is Research Towards Control of the Cassava Whitefly, Bemisia tabaci.
Sorry, just to interrupt you, you've moved off presentation mode. You want to move back onto presentation mode, please? Thank you so much. Great.
Okay, it's now in presentation mode.
Perfect. Yeah, that's first slide. Thank you so
much. Oh, good. Okay, thank you. Okay, so the title is Research Towards Control of the Cassava Whitefly, Bemisia tabaci. And I start with a little introduction of cassava. For those of us who are in Africa, we know it's a semi-perennial root crop, and we use the root and both the leaves for food. And currently, it's becoming kind of commercialized, and it's being used for several industrial purposes. But one major important aspect of the cassava, especially with the climate change, is that it's a food security crop, which is tolerant to drought. And now it's viewed as one of the crops that will boost Africa when it comes to adaptation to climate change. Okay, so Africa, we are the leading in production. We can see from the chart here. But then you find that when it comes to the yield, we have the least in terms of production, because then there are several factors that lead to low yield. And one of those factors, we all know that apart from poor agronomic practices, we have biotic factors that are not found in other continents, such as the Asia and South America. So a little bit about the cassava whitefly. First of all, it's under the Bemisia tabaci species. And that Bemisia tabaci species is a pest to over 1,000 hosts and mainly causes damage by sap-sucking and transmits well over 300 plant viruses. In Africa, the greatest damage done by this pest is the transmission of cassava mosaic disease and cassava brown streak disease. And yield loss estimates are about 50%, which account to almost 1.9 to 2.7 billion USDs annually. That is the combined damage of the whiteflies and the viruses. Okay, for cassava mosaic disease, we know that it's caused by cassava mosaic begomoviruses. So far, there are 10 known species, and Africa has about 8. So the symptoms are very obvious most of the time, causing mosaic, mottling, and leaf distortion, and severe crop standing. Then the second disease, which is cassava brown streak, that one is caused by epomoviruses. And so far, there are two known species, cassava brown streak virus and Uganda cassava brown streak virus. And cassava brown streak disease is so far restricted only in East, Central, and Southern Africa. It hasn't reached West Africa or any other parts of the world. And the symptoms are mainly leaf mosaic. Then we have root necrosis and stem dieback. So I'll talk a little bit about now the genetic diversity of the Bemisia tabaci. When you say the cassava whitefly is under Bemisia tabaci has over 40 cryptic species. And the cassava group is one of those cryptic species. You can see it was classified as the sub-Saharan Africa. And it's here, as you can see, compared to the so many other cryptic species that are found across the world. And these cryptic species are mainly found on other crops. You'll find them on sweet potato, on tomato. You'll find them on maybe eggplant and so on. But so far in Africa, they do not colonize cassava. When you put them on cassava, they will die. So the species on cassava is specifically on cassava. And one characteristic of the Bemisia tabaci is the cryptic species respond differently to host plant resistance, to insecticide, and to virus transmission. So the cassava has kind of adapted specifically to cassava. You can find it on other plants like sweet potato, bean, soybean, but mostly it's on cassava. So we did a little bit study where we wanted to understand this cassava Bemisia tabaci. Originally, they are characterized mainly by sequencing the mitochondrial gene, but that is very limited because it's only one gene. So this map was produced after doing SNP genotyping. And so now the cassava whitefly has, the cassava Bemisia tabaci whitefly has 6 major haplogroups, and you can see they are distributed according to the region. So we find that these two, WA and SSA2, we find them mostly in West Africa and a little bit in East Africa. SSA4 is restricted in Central Africa, mostly in Cameroon. Then we have a small subgroup here, SSA-CA, that was in DRC. Then we have SSA-ESA, which is mostly found in Southern Africa, part of East Africa. And this red one here is the SSA East and Central Africa. This, as the name suggests, is found in East and Central Africa. And it's the one found in the region where we have high epidemics of CMD and CBSD. And the reason why it's associated with such high epidemic is because it reproduces in super abundant population. And so it is the species of interest when it comes to epidemics of cassava viruses in East and Central Africa. So this chart just summarizes a little bit. It's a survey that was done in Kenya, but I just wanted you to see how these whiteflies can be super abundant on cassava. You see, these are the number of farms that were surveyed, and you see whiteflies ranging from 0. Then you find where you find like 0 to 10. Those are on 5 top leaves. Then you come across the ones that are ranging between 10 to 100. That's already high. By the time it reaches 100, it's already being classified under super abundant. You have farms that are having 100 to 200 whitefly, and then over 200. And basically you find these plants are just kind of overwhelmed. You could observe some are wilting because of the heavy infestation. I mean, whiteflies, they cause damage already by sucking sap from the plant. So if the population is very high, the plants will get stranded. So we asked ourselves, why do we need to control these whiteflies? And the first point is, previously, whiteflies have been ignored because cassava has been mostly a subsistence crop where the farmers just did with the little they could get. They didn't bother to invest a lot. But now it's rapidly evolving from a subsistence crop to a commercial crop, meaning farmers who have inputs can at least be able to invest in the crop to get maximum yield. And then the second reason is we know that it's a vector of viruses causing CMD and CBSD. And then the third reason, we find that they have introduced a lot of tolerant or resistant varieties. But despite all this, CMD and CBSD is still a great challenge to cassava production in sub-Saharan Africa. And then the other challenge is that the whiteflies are a threat to the success and sustainability of the clean seed system. Recently, there have been efforts for the past, I would say, almost 8 years now, efforts to have a clean seed system in sub-Saharan Africa. That has been pioneered in Nigeria and Tanzania under the seed system. And the thing is, if the clean seed is going to be given to the farmers and the whiteflies are out there, then it means that the material will just get infected. In most cases, it just takes one season. You give a farmer clean seed, and if the whiteflies are there spreading the viruses, by the end of the season, it is 100% infection already. Then the last one is that observing from the damage this whitefly causes on cassava, it's already a pest that will cause damage in the absence, even in the absence of viruses. And when we look at climate change, plants are going to be subjected to various stresses, water stress and drought. And then, okay, the last thing you will want is a pest already sucking sap from plants that are already under water stress. Okay, so now our research mainly at IITA has been trying to focus on how we can control these whiteflies. And then I'll start the introduction a little bit by talking about the IPM pyramid of tactics. So we always say that we start with cultural practices, maybe getting clean seed, planting resistant varieties, then maybe removing infected materials and so forth. So of course there is effort to have the tolerant varieties and clean seed, but the effort to get the farmers to remove the infected material is still challenging. Then we have the physical mechanical. Of course, that cannot work with the whiteflies. Maybe in a vegetable production where you may have shades or screen nets or such thing. Then we have biological control where we have like natural enemies and entomopathogens, mostly the parasitoids that control whiteflies. Then we have the biopesticides. These ones have been formulated commercially. Find like they could be botanical oils or they could be entomopathogens. Then at the top, we have now the synthetic pesticide. So our research at IITA has mainly focused on like trying to see biological control, then biopesticide and pesticide. So I will say we have focused on testing chemical insecticide, and we have some entomopathogens and botanical oil. So the first, I will start with the insecticides part of it. We started with what we refer to as a novel insecticide. I mean, from the studies reported, it's not very harmful to natural enemies and to the bees. So then we carried out studies. Mainly, we started with the lab work where you can see we were screening in the lab. This is a lab experiment where you dissolve the insecticide in the water. Then we introduce whiteflies to feed. This one we were using the leaf, and then we ended up doing with the cages. And then we proceeded to the field where we did like spraying as recommended by the manufacturer in doing the cutting dipping. The cutting dipping is where you prepare cuttings, dip them in the insecticide just once, then you plant. So my presentation on flupydifron will focus mainly on the cutting dipping. The reason why we are interested in the cutting dipping, because one thing we know that farmers may not afford the regular spray, like spray every month or maybe every 2 weeks. Then another factor is we don't want to harm the natural enemies out there. I mean, we all know that these insecticides are harmful, not only to human health, but to the very natural enemies that we need to be preserving so that they aid in the control of whiteflies. So this is an experiment where we did the cutting dipping. We just prepared the cassava cuttings, 20 centimeters long. Then we dip them in the insecticide. So we did an experiment with cassava farmers in Mukuranga Coast region in Tanzania. And the variety was Kiroba. So we just had like 400 plants per treatment. The cuttings were dipped for 1 hour in flupydiflon. That was the concentration. And then after planting, data was collected for 6 months. And the data mainly collected was on whiteflies, CBSD, CMD. And then later on, at 10 months after planting, we did yield, which was root number and weight in kilograms. And these are the results that came out from that study. You find this shows the adult whiteflies. You can see that they were suppressed by an average of 40%. And the nymphs, which were counted on the 9th and 14th leaf, were suppressed by 65%. So in this data, we see that like monitoring nymphs can give a more clearer picture of how the insecticide is reducing whitefly population in the field as opposed to adults which just fly around. And when it came to the disease incidence, we have CMD reduction by 16%. And CBSD, you can see that CBSD, the reduction is very low, 2%. And you can see what these whiteflies can be up to. All these plants were healthy. At the beginning, they were clean planting material. But because of high populations of whiteflies in the study area, you see that by the 2nd month, 3rd month, the plants are already having CBSD. And then, so the insecticide may not be good at managing CBSD because it's a semi-persistently transmitted viruses. So when we go to the results on the yield, you can see that the root yield, this was in grams per plant over here. Then we have the control here, and then the ones that were dipped in flupydiflon. And see there was an yield increase of 49%. Then we have the number of roots. The number of roots increased by 38% in the treated. When we check the CBSD in the root, it was very low. That was less than 2%. But it's because Kiroba is a variety that is tolerant to CBSD. So then another study we have been carried out was the flupydiflon one. We tested it, but then the insecticide is not yet registered in Tanzania. So we embarked on testing another product. We refer to it by the name MandiPlus. MandiPlus was formulated by Syngenta Foundation for Sustainable Agriculture. And so they mix the insecticide Cruiser, which has thiamethoxam, and then the fungicide Maxim XL, which has these two compounds, fludeoxonil and metalaxyl. So to make the mix of MandiPlus, like for 100 liters of water, one will need to take these ratios. And then they include the white vinyl paint. This white vinyl paint is just like included for a warning purpose to make the solution reddish. So this is what we did with the MandiPlus in research plots. So we had a research plot in Chambezi, which is not far from Dar es Salaam, where I'm based. And then we had like 30 plants treatment per plot in 4 blocks. The variety was Kiroba. And the same thing, just cuttings dipped in the solution, then planted just one. And then we collect data at 1, 3, and 6 months after planting. on whiteflies names and CBSD and CMD, and the yield on root number and weight in kilogram. And you see from the results, okay, we are having like different products that we set out to compare alongside that MandiPlus. So we'll have MandiPlus, and then we have imidacloprid, which is the control. And then we have Lecatec. Lecatec is an entomopathogen, which we just applied by spraying on a monthly basis as per recommendation. We wanted to see whether it has any effect. Then we have imidacloprid, we combined with Lecatec. And then we have MandiPlus combined with Lecatec and the control. And you can see from the results, the adult whitefly, you can see the significant effect of MandiPlus, where you see that MandiPlus ended up reducing whiteflies by an average of 82%. And here MandiPlus does reduce names by an average of 85%. And then it comes to CMD, you see that it drastically reduced the CMD incidence by 89% compared to the control. And then we have the CBSD, which was actually also suppressed by MandiPlus by up to 55%. When you come to the yield of the MandiPlus cutting, you see that the MandiPlus, which is here alone, had yield increase of 53% compared to the control. And then this was MandiPlus sprayed with the Lecatec, the entomopathogen, the MandiPlus with Lecatec is here, which increased yield by 67%. So by that, based on the estimates of the yield that we got from the MandiPlus plots, if we multiply by the 4,000 or 10,000 plants per hectare of cassava, you find that that will be almost be estimated at 60 tons per hectare as compared to 39 control. In these images, you can see the cassava that came from the control and the cassava that came from the MandiPlus. So another study was to do the entomopathogens. We started with testing out some commercial products. We have mycotol here. That is the scientific name of the entomopathogen, Acanthamyces lecani, and then Acanthamyces lecani for Lecatec, and then Acanthamyces muscarius for mycotol. So these are commercial products, but the isolates are not from cassava ecosystems or Tanzania. This one was from Europe, and Lecatec is from Kenya. So we did out like experiments in the field to test how they perform. And then so we set out the experiments in Chambezi and Mkuranga and sprayed on monthly basis, then collected the same data on whiteflies and cassava viruses, and then yield at the end of the 10 months after planting. And you can see there is still a trend there. They don't do much for the adults. Of course, they reduce the nymphs because they attack the nymphs. Entomopathogens are tailored to attack the nymphs, and rarely do they attack the adults. So we find that there is a reduction in the number of nymphs. When it comes to the virus diseases, unfortunately, they did not show any significant effect suppressing the viruses. Same pattern from Mkuranga location, only the nymphs were significantly affected. Then we go to the yield. You can see that the root yield was also not significantly different at both locations for the Lecatec and mycotol. You will observe that the CBSD incidence was very high, yeah? But it's because the variety tested is Albert. Albert is a variety that is susceptible to CBSD. So if you look at those two comparisons, Kiroba having less than 2% root incidence and Albert with 80%, that already highlights the importance of host plant resistance in management of these viruses. Another study we have been doing is to test the botanical oils. So we started off with the lab experiment and then proceeded to the field where we tested neem, a crude neem oil, and then we had a commercial product, top neem super. We just wanted to see how these botanical oils can affect, or if they have any effect on whiteflies. Then we set out the research plots. We did the spraying on monthly basis and then collected the data on viruses and then whiteflies and root number in the yield. And from that, you notice that the neem products were able to reduce the whitefly numbers. And they also reduced the neem 63 to 70%. And then, of course, you can see that when it comes to CMD and CBSD, the effect is very minimal. But one interesting pattern we see here is they tended to kind of delay somehow the onset of CBSD up to almost 4 months. You know, of course, oils have been reported to interfere with transmission of persistent and non-persistent viruses. So maybe that is something we can look at into the future. Oh, when it comes to the yield, of course, number of roots, we don't see much difference. But when it comes to yield kilograms per plant, we see that at least the neem crude oil had a significant increase of 55%. So then the reason why we have had the neem and the entomopathogen is we are trying to establish like an IPM where you are having several aspects aimed at controlling the whiteflies. So our thought is that the farmer can do the cutting, dipping in an insecticide plant, and if there is need, instead of them like spraying insecticides again, they could spray entomopathogen or botanical oils, which are less harmful to the environment and to the parasitoids and predators. So now we have an ongoing research. This is being funded under the CECTEL. And then, so we have set up a research for farmer participatory trial. This is very critical because all that are presented was done mainly in research plots. So our aim is to find out, does it work under a farmer condition? And we have set up a participatory trial where we are evaluating the efficacy of Mandiplast with 10 farmers in the Chato Lake Zone, a map of Tanzania. And you find the Chato district is here, and this is where the farmers are. So how we set up this experiment is we had farmers volunteer 2 separate pieces of land, like 1 acre and half an acre that were separate. And the reason why we are doing this is because in research plots, you can see from the results I've presented is that even in treated plots, there is still impact of viruses. It's because the research plots are so small, the whiteflies just keep moving around freely. But now with the farmer, we are going to use a bigger plot, and then the control will be separated from the treated plots. So the 1 acre is treated where we have 4 sections, each with 1,000 cuttings. In those 4 sections, they have the Mandiplast, then we have the imidacloprid, this is the control insecticide, and then we have Mandiplast where we are going to spray Lecatec and imidacloprid with Lecatec. Then the half acre is controlled with no spraying. So the experiment is currently going and data is being collected at 1 month. We already have data for 1 month, and then we will collect data at 3 months, this end of March, and at 6 months. Then we'll do root yield at 10 months after planting. So then we are also evaluating some botanical oils from native plants. We have, apart from neem, we have others that are common in farmers' field, like Tagetes, Tephrosia, and Tithonia. And from those results, we can see that these other botanical oils are also promising. They perform just as good as neem, especially the Tagetes and the Tephrosia. And we also have, we are bioprospecting for novel entomopathogens in the cassava ecosystem. Because we know that, okay, the entomopathogens we tested are commercial, not native isolate. So we want to bioprospect from the cassava ecosystem and find out if we can get any isolates that are adapted maybe to the cassava whitefly better than the other whiteflies. But so far we have some isolates. You can see that, okay, from the preliminary results, what we have found is still not performing as good as the mycotol. But of course, mycotol is commercialized and I would say optimized for use under the field. These are just like raw isolates from the soils. But the process is ongoing and we will do more tests to see how far we can go with this. So with that, I will have my concluding slide where we can say that cutting dipping in insecticide effectively reduces cassava whitefly. And you can see from the result that it increases root yield. Okay, the commercial entomopathogen Lecatech, this was under the research plots. It does not seem to show any significant gain when combined with cutting dipping. But then we see what comes out of the participatory trials from the farmer trials to make the final conclusion on the entomopathogen. And I'll say that this experiment that is going on under the farmer participatory trial will help us determine the efficacy of this cutting dipping in Mandiplus, whether it is effective under farmer conditions. Then, of course, we are doing the bioprospecting for native isolates of entomopathogens, as this could be more adapted and more lethal than the imported product. So then with that, I will say that most of the work that I've presented here is just for research purpose. It's still ongoing research. Nothing has been recommended to the farmers yet. But currently with the Mandiplus experiment with the farmer participatory trial, that's what we are working with the Tanzania pesticides, the Tanzania Plant Health and Pesticide Authority. So if the results come out well and convincing, that's when they'll be able to authorize the use of Mandiplus by cassava farmers. And now that means after that, we will need to embark on like scaling it out to farmers. With that, I would like to acknowledge mostly the donor and all the collaborators and all those who have contributed the success of this work. Thank you.
Great. Thank you so much indeed, Dr. Wosula. That was really, really excellent. And as our first presentation, you really set the bar very, very high. So we really appreciate that very much. Over 90 people attended, so that's really excellent. We have a number of questions already. Stephen Walsh from USAID had a question. Stephen, would you like to unmute yourself and ask, please?
Can I stop sharing, David? Or...
Yeah, please stop sharing. Thank you so much.
Oh, thank you. Yeah, okay, good. Yeah.
Thanks so much. Yeah, over to you, Stephen.
Yeah, thank you very much for the presentation. Excellent work. I guess the number one question for me is just to try to understand the difference in effectiveness of the Bemisi tabaci to spread CMD versus CBSD. My understanding is that it's much more of an efficient transmitter with cassava brown streak versus cassava mosaic. So I'm wondering if you might be able to talk about any additional research on that. And then I guess my other point was just under the excellent slide you presented with the control mechanisms, IPM, at the bottom of the pyramid was cultural practices. And I just sort of wanted to note that, you know, I know you're already aware of this. IITA has done some fantastic work on identifying regulatory opportunities in terms of working on different protocols for assessing the quality control of cassava seed fields. So I appreciate hearing some of your feedback on these points. Thank you.
Okay, thank you.
Would you like to address that question, Everlyne, on the differences with CBSD?
Oh, okay. So now for the transmission of CMD and CBSD, yeah, of course the whitefly is more of an efficient transmitter of CMD because CMD is persistently transmitted. Once the whitefly is acquired, it will continue to transmit it until it dies. But CBSD on the other side is the non-persistent. And then so one thing semi-persistently transmitted. And one thing we have observed actually under like research work where you have to do transmission of CBSV and UCBSV using whiteflies, like putting them in the cages and so on, it is very, very difficult. In fact, the transmission rates are very low. But under the field conditions, the transmission rates are very high. And the factor that makes the whole difference is the abundance of whiteflies. If those whiteflies are so many out there, it's like that mass of them is what causes the transmission of the CBSD go up. And that we have proved by a trial we already did, like in the southern islands. The southern islands of Tanzania, we did a trial there where the whitefly populations were very low and the incidence of both CMD and CBSD were very low, below 10%. And interestingly, the type of whitefly in southern islands is naturally the one that doesn't reproduce rapidly. If you saw the map that I showed of Africa, of the 6 haplogroups, that haplogroup in the southern region, the population are not so high. The problem comes with that group in the Eastern Central Africa, where I highlighted red. The populations are so abundant. And that's why you see the challenges of CBSD over there as compared to like other parts of Africa.
And just for clarification for the audience, if it's semi-persistent and it is not the case that it's just inside the mouth parts, the proboscis, and as the whitefly goes from one plant to the other, it quickly infects. Is that the case? Could you explain the difference between persistent and semi-persistent transmission?
Okay, okay, good. Yeah, of course, when it comes to like the semi-persistent, of course, the persistent, they say that the whitefly has to acquire the virus, and then it has to go from the saliva to the salivary glands. So then from there, it will keep being released as long as the whitefly is alive. It is in the salivary glands, it will keep being released as long as the whitefly feeds on the plant. That is for persistent. But for semi-persistent, which is under where CBSV falls, that one, the virus is not retained. In most cases, it's just obtained by the whitefly through the mouth parts. Sometimes it can be just retained around the mouth parts. And then it will be spread like within 25 seconds to minutes, it can be transmitted. But then the whitefly won't keep carrying it forward. Like it will feed on an infected plant, and in seconds or minutes, it has already infected a healthy plant, and that is it. And that's one reason why, like controlling such viruses, the semi-persistent viruses, using insecticides is very difficult because you can spray the whiteflies, yes, but they will only have seconds or minutes to infect a healthy plant. But for persistent ones like CMD that take hours, then it's easier because once you kill an infected whitefly, then that is it.
Great. Thank you so much for that clarification. And next question is Dr. Matthias Tembo from the Zambian Agricultural Research Institute. Matthias?
Yeah, thank you, David, for this opportunity. And thank you, Everlyne, for the nice presentation. Wonderful work you're doing. My take is looking at what you indicated with regards to the species diversity, and then in addition to the CMD and CBSD diversity across the different regions. I would feel there could be some little bit of issue with responses to the pesticides that you are looking at, that you are working on, that you are researching on. And I'll suggest, if it's possible, that at the same time you're doing it in Tanzania, whether this can be replicated to other countries as well, looking at the diversities that could be also be found in the different countries, vis-a-vis CMD diversity, CPST diversity, and the whitefly species diversity. I don't know what you would be your take on this one.
Okay, thank you, Matthias, for the question. Yeah, actually, of course, we know that they are doing it in other countries is very possible. All we need is get funding. But this participatory trial we are doing will be the best. So because once we get the result from this participatory trial, then what goes to other countries, like if you want to test in Zambia or Uganda, they've already done this kind of testing. We'll maybe want to test in Kenya and maybe other countries where there are problems of viruses and whiteflies. So our aim will most likely go for the participatory trial. Once we have the Tanzania participatory trial results and prove that it's effective, what we do in other countries will just be participatory trials instead of starting all over again with research plots. So that is very possible.
And Matthias, this is one of the functions that we're trying to achieve in the Innovation Lab, Current Emerging Threats to Crops Innovation Lab. We want to have these research trials happening in multiple countries at once, so that we can test the variety of conditions, cryptic species, which might vary, agroecological conditions that might have an effect, or farmer races that are preferred. And so having all of these studies happening, but the problem that Everlyne identifies is cost. And so one of the things we're trying to do is massively bring down the cost of such trials by leveraging local young people from universities across the continent. And that's an important component. So in the Innovation Lab, we have 18 graduate students in 6 universities. And then we have a lot of field officers, and this will be an effective way to do that. And that means that experts like Dr. Wosula can have an overview of this, but just be able to collect data at scale in parallel, and also people like yourself. So thank you so much. Okay, so let's go to one of those young people, Agnes Kampire from PlantVillage, Uganda. You had a question?
Thank you, Everlyne, for the presentation. Among the biopesticides, you talked about neem oil. I wanted to clarify. I wanted you to clarify about its efficiency. Like how much do I have to apply, for how long? Is it from planting up to the period of harvesting?
Okay, thank you for the question, Agnes. So for what I'm presenting here is, as I mentioned, is like still under research. It has not reached a level where we go out and tell the farmer that, okay, you apply this kind of neem and apply it at this amount. But from the study here, we were testing, it was 1.5%. But that is like the crude oil where we just bought the neem oil and we were mixing it with the ethanol 4%. But the commercial products out there, they have their own formulated products with instructions on how to apply. So if I may ask, were you interested in like using neem or Agnes?
Of course, there's a lot of cassava whitefly infestation in Uganda, specifically Eastern Uganda. So I thought it was an easy procedure to carry out, but good enough, you said it's still under research. It's not yet confirmed.
It's under research. I mean, when everything is done, when we come up with the recommendation, that's when you can go out and tell farmers, okay, this is what you need to be doing. Yeah. But for now, it's still under research until we have all those. You see, we are doing like the farmer. We have started with the farmer participatory trials with Multiplus. And then that works fine, then it is approved. Then now you can go out and train farmers and tell them, okay, now this can be used. But for now, all those are just like under research in progress.
Great. Thank you so much indeed. So then another one of those students I mentioned, Tracy Jo from PlantVillage Kenya, you had a question? I can't hear you, Tracy. You're not muted, but I can't hear you.
Can you hear me now?
Yeah, perfect. Go ahead.
Yeah, so thank you, Dr. Everlyne, for the wonderful presentation. It's an amazing work you're doing at IITA together with your team. So my question is, are there any research that is going on if these biopesticides or botanicals have an effect on the nutritional quality of cassava? Like, do they interfere with the original tubers that come out of it, or it's just the yield we are focusing with the yield that you guys measured? Are there any tests on the nutrition effect from the botanicals, from soaking the botanicals in the, yeah?
Oh, okay, thank you, Jayo, for the question. Okay, so I would say that for the botanicals, they are not absorbed. You know, like their effect is contact. It's never systemic, so they are not absorbed by the cassava plant. That means they don't have any effect on the nutritional value of the cassava. And I would say that there's no evidence where it is indicated that pesticides can have effect on the nutritional value. The only thing is probably you will say if they are being retained in the plant. And that one for the cutting dipping, where we are dipping insecticides, with the farmer participatory trial, we are going to test, are they still present in the cassava plant? Because you can see from the results, they suppress whiteflies for a long time. So the question many will ask is that, could they be still in the plant? So that is what we are going to test, and then we will present the results.
Great. Thank you so much indeed. And then Nancy Onyango, also from PlantVillage, Kenya. You had a question? Thank you, Nancy.
Okay, thank you for the wonderful presentation. My question was, just a minute, I wrote it down. By the use of insecticides, do you think it kills the adult whitefly like 100% by the use of insecticides?
Okay, thank you, Nancy. Okay, so now you can see from the result, you can never kill the whiteflies 100% because they are always reproducing. They will reproduce on other plants, so you can never eliminate them 100%. But what we want to do with the use of insecticides is bring the population down. You can see that their infestation on the plant already causes yield reduction, and on top of that, they are transmitting the viruses. So bring the population down to a certain threshold where they are not damaging the cassava and where they are not transmitting the viruses rapidly, so that we bring up the yield of the cassava. So that is the aim.
Great. Thank you so much.
Thank you.
Finally, maybe the final question from Salome. Kwagala in PlantVillage, Uganda. Salome?
Yeah, thanks. Thanks, Everlyne, for the presentation. So during your presentation, I noted that Kiroba variety is actually resistant to CBSD, and then the dipping in the chemical caused an reduction of CBSD in like was it 40%? So I'm wondering, how do you like account for that? And then my next question was, there's a treatment which delayed spread of CBSD for like 4 months. Yeah, I think 4 months. And then have there been any studies to show maybe if we can take advantage of that treatment, majorly for areas that are highly infested with whiteflies and where CBSD is a very big issue? Thank you.
Okay, thank you, Salome, for your question. So the first one, you asked about the reduction of the CBSD by about 40% in the variety Kiroba. Okay, Kiroba, the variety itself is tolerant to CBSD. So the incidence we were scoring is by looking at the leaves, like the plants itself. You look at the leaves and you just score that, does it have CBSD or no? Then you calculate that incidence, like the percentage. If I have 30 plants, how many are showing symptoms? But the root incidence of Kiroba, the one I mentioned of 2%, is the root. You know, the most critical part of the CBSD is the root. You could have the symptoms on the leaves, but the root is still okay, then that is fine. The greatest yield loss is when the root is damaged. So Kiroba is tolerant, that's why it had about 2%. But the other variety, which is Albert, you saw that it had about 80% incidence in the roots. You saw that the roots had CBSD symptoms. That is one is very, very susceptible to CBSD. Then, of course, you saw a treatment that is on botanical oils, where the reduction of CBSD was up to like 4 months, which was kind of surprising because CBSD is the one that usually picks up very fast. In fact, I only observed that with the botanical oils, not with those other treatments. And that was just a one-time treatment in the field. We are doing the second round, and that we'll see whether that trend is still there. But oils generally have been known to suppress the transmission of persistent and semi-persistent viruses. So we don't know whether that could be an effect. It is something we'll look into further.
David, you're muted.
You are muted.
Thank you so much. I was just saying we're going to close up, but we just had some conversations in the chat there regarding tolerance and resistance that people can see. So we will put all of this together. We will make available the presentation on YouTube for people to watch later on. We'll also make available Dr. Wosula's slides as well. And we're certainly happy to have a follow-on seminar on this topic. There's a lot of people working on this extremely important area, but Everlyne, this was a fantastic talk, really a great attendance and a huge amount of questions. So thank you so much indeed for taking the time to do that. We really appreciate it. Everybody, thanks for joining. And clearly this has started off well, so we'll do lots more of them from the Current and Emerging Threats to Crops Innovation Lab at Penn State. Thank you all very much and enjoy the rest of your day or evening wherever you are. Thank you, everybody. Bye-bye.